have you here, and your talk brings in many current and important themes. And so you have about 45 minutes. So please, thank you very much.
Thanks, Robert. Thanks for the invitation today. And it was really great to see all of those flash talks from the students, especially. Can you hear me okay? Okay, good.
Yep.
So like Robert mentioned, I'm part of the Luffer Center, and I'm really looking forward to being back there. We share a building with IACS, for those of you that don't know, and it'll be great to be back in the same building again. So today I'm going to talk about COVID-19. I'm sure I don't need to give a lot of background on this. COVID-19 is caused by a virus, a coronavirus called SARS-CoV-2. And there are different coronaviruses. This is not the first that we've had that can infect humans. The common cold, there are several coronaviruses that caused that. There were the SARS and MERS outbreaks, and then, of course, COVID-19. And these seem to keep getting worse, and this is not certainly the last coronavirus that humans are going to have to deal with. And so what I'm going to focus on today is whether we can have high-performance computing help us prepare for the next pandemic. So I'm sure that lots of you have seen pictures of the virus. They're called coronaviruses because they have these spikes coming out of the surface, which in these micrographs, you can probably just barely tell that there are these projections out of the surface. And all of these make it look like it's got a crown, and that's why they're called coronaviruses. So zooming in a little closer, this is very small. It's 100 nanometers in size. And it would be great from experiments to get a better idea of how this virus worked and how all the moving parts work, but we can't do that really. And so what we're going to do is substitute our computer modeling. And so this is a model of the virus. They tend to have, we know from experiments, about 25 of these spikes sticking out of the surface. I'm going to zoom in on one of them. There are trimeric proteins, so there's 3 identical proteins that make it up. And in a lot of my pictures, I'm going to color those with red, yellow, and blue. There's a light part that's on the top and a dark part on the bottom, and I'll talk about those in a little more detail. And so whenever I'm showing one of these, imagine that this is what's on the outside surface of the virus. Okay, I know a lot of you aren't biologists. You're probably thinking to yourself already, I'm lost. I don't even know what a protein is. So if you do hear about proteins, usually when we talk about them, we're talking about diet and how you need protein in your diet. There's lots of proteins in different foods that we eat. It's become very popular now for food items to be labeled with how much protein they have. And the reason that you need protein in your diet is because you need these as raw materials to make your own proteins. It's kind of an ideal case of recycling. You eat these egg proteins or nut proteins, and your body takes them apart and puts them back together into human proteins that you need. And so we're going to do a little bit of molecular biology to get people up to speed. I promise not very much of this. All cells have DNA in them, and this stores your genetic code. This is basically the blueprints for all of the proteins that you need to make. All of these ATs and GCs that you might have learned about in biology class, those are just the code for how to make proteins. That gets turned into messenger RNA. The DNA still stays in the nucleus, so you make messenger RNA that goes out, and it interacts with something called a ribosome that uses that code and turns it into these long floppy chains of amino acids that are called proteins. And then those floppy chains end up folding up into a pretty well-defined shape, and that determines their function. And proteins are involved in all the different machinery in your bodies. They store things, move things around, do the chemistry, many things, including being parts of viruses. Now, this is a glycoprotein. On the surface, there are all these little gray things. You can think of that as being sugar-coated. So this protein is like a peanut M&M with a candy coating. Okay, so this lets us talk about the vaccines that are available, and they are all trying to expose your body to this spike protein for reasons that I'll get to. There's DNA versions, like the Janssen J&J vaccine gives you DNA that lets your cells code for the spike. The Moderna and Pfizer BioNTech ones give you mRNA that does the same thing. It lets your cells produce spike protein. And then the Novavax one that may be approved here in about a month or so, that one actually gives you spike proteins. And so all of these vaccines are doing the same thing, which is to expose your immune system to that spike protein. Why does the spike protein matter and recognizing it matter? Because it's on the surface of that virus and it unlocks entry into the human cells or to whatever cells the coronavirus is going to infect. So here's our spike over on the left. It's sitting on the viral membrane. And here's our host cell. And in the case of SARS-CoV-2, it interacts with a molecule called ACE2. and that's on the host cell membrane. And the goal of the virus is to get its genetic material into your cells so that you start making viruses. It can't reproduce on its own. And that's kind of a complicated process because you've got your own membrane that tries to keep it out. And so the first step is that this spike binds to the ACE2 receptor. It forms a complex that's specific. And then the next step that we don't understand too well is that a big chunk of this, called the S1 subunit at the top, like the cap of the mushroom, it's going to come off and stay with the ACE2. And now we're left with just the stem of the mushroom behind, and that's called the S2 subunit. Now that stem of the mushroom is unstable, and so it's going to release these things called fusion peptides that are like harpoons. They're going to attach themselves into the host cell, and then this thing is going to refold itself and pull this viral membrane up, merge them together, and that lets this virus into the cells. And so all of these steps of change are the key to getting that virus into the cell. And if we could block that, then you wouldn't be able to get infected by this virus. Now, I've drawn these. These are actually pictures from coarse grain models that we do simulations on. It's important to keep in mind that these two steps are just models that we built based on ideas. From experiments, we don't really have any idea what happens between interacting with ACE2 and this final structure where the membranes have merged. We don't know how that works. And so that's what we're going to try to get out, some simulations. So the spike is flexible and changes shape a lot, like you saw on that last slide. And one of the important parts of that is that there's sort of an opening and closing event. This one's called closed. The mushroom top is all packed down. And then in order to bind to ACE2, it has to open up and form this open state, and then it can interact. If we try to take that ACE2 and kind of dock it back into where it would go in the closed structure, it doesn't fit. It's got an overlap there, and so we know it has to open. It can't be closed. So if the whole job of this spike is that it's supposed to bind to ACE2 and recognize it and let the virus into the cell, why would it ever close? That seems really counterproductive. It should just be in the open form all the time. And the reason that it closes is because antibodies also bind to this spike. And so your immune system is always on the lookout for things it doesn't recognize. And so I'm showing you here on the right, antibodies bound to that same part, what's called the receptor binding domain. They bind to the same place as ACE2. And so the virus, being sneaky, doesn't want to have that exposed. It would rather not have antibodies bind to it and keep it from doing its job. And so it closes down. And when it's closed, it can't bind to ACE2, but it also can't get stuck to those antibodies. And every now and then it just opens up with some rare transition, checks to see if there's an ACE2 around, and then closes down. Most of the time it's closed to protect itself. Okay, but antibodies do bind, and hopefully most people have gotten their vaccines now. I just had my second dose last week, and this to me is one of the biggest successes of science in history. It's really amazing that within the course of a year, we were able to not just develop these vaccines, but test them and distribute them and get them into people and stop things. And so this is amazing. But as you may have heard, there's still reason to be a little concerned, even with the vaccines, and that's because the virus is evolving. And so we have these variants that you hear about a lot in the news. They call them things like the UK variant, the South American variant, things. They've got these numbers, like the B117 variant is what was first identified in the UK. And these variants so far do not seem to overcome the vaccines, but they do reduce its effectiveness. And it's probably likely that within a year, we're going to need booster shots so that we can deal with the new variants, and the current vaccines may not be as effective. And so what is the virus doing when it evolves? Well, there are mutations that are made. One of the common ones that you see in lots of variants is called the E484K mutation. And that just means one of those amino acids out of a few thousand in this spike has changed. And so what does E to K mean? Well, E is the shorthand for a glutamate, which this is the chemical structure of that amino acid. It has a negative charge on it. And K is a lysine, as it has a positive charge. And so what the virus did was it substituted a negative charge on the surface for a positive charge. Charge complementary is really important in recognition and interactions. And so the antibody comes along expecting there to be a negative charge, and now it's positive, and instead of being attracted to it, it's repulsed. And that's why these variants can be so challenging. So this is a problem in general with antibodies. They're very big molecules, and they can only bind to the surface. They really can't get into much other than the surface. They recognize the surface shape and the chemical color. So you can imagine that if I gave you a task that you're supposed to recognize cars and stick to them so they can't drive around, here's a bunch of different generations of Honda Civics, and you would look at the first generation and you'd say, okay, it's blue and it's got narrow spikes on the wheels. I'm going to recognize that next time I see it. And that's what your immune system does. Next generation comes out, it's not blue anymore. You have to retrain. And so it says, okay, but it's still got those narrow spikes on its wheels. That's what I'm going to recognize. Uh-oh, next generation, they changed the wheels. You don't recognize it anymore. And now you say, well, there's a black stripe, and that's got all of them. Next year, no black stripe. And this is the problem with your immune system, is that all of these things on the surface don't really have much of anything to do with function. They're just decoration. They don't do anything. And so the designers can swap them out, and it doesn't really matter. Now, knowing about cars like we do, you would say, well, it's easy. Pop the hood and disconnect the battery. That's going to work on all of them. Or make it so that you can't open the doors. That will work on all of them. But that means we need to understand function. And we don't understand function here. All we understand is shape. And so what we need to do is find out how the machine works, understand what the important pieces are, and then presumably those pieces are something that the virus can't swap out because they're key to function. And it wouldn't be able to get into cells anymore. And so what we're going to do is we are going to use computing in order to figure out function. So I won't get into a lot of the details, but if you're interested, a few of us in the Laufer Center last year wrote this review that highlighted in part our work on the spike and then some other stories from the field over the past few years that give you kind of an overview from a non-expert perspective of how computing is working in molecular biology. So that's the story I'm going to tell you today. I'm going to give you a little bit of a background on how we do these, since this is a computational science group. And so the basic problem is that experiments give you averages. We average over lots of time, we average over lots of molecules, and when we look at averages, if we look over long periods of time, like with a camera on slow shutter speed, we see things get very fuzzy. They're blurry because we're averaging over motion times and we don't get a clear view. As the shutter gets faster and faster, we start to get a very clear view, but now we lose motion. And so it's very difficult to get both from an experiment where you can understand all of the structural details in high resolution, but you also have time resolution. And so that's what we try to do with our simulations. So we typically start with a structure that is an average, like this one here, a high resolution structure of a protein. We often use these ribbon diagrams to show us the overall shape of the polymer chain. And so you see like these purple helices and these yellow sheets, and they just tell us sort of how it's organized in space. And that's great. These experimental structures are wonderful, but what we need to understand is flexibility, especially for flexible molecules like the spike. And I really like this quote by the Nobel Prize winning physicist Richard Feynman: everything that living things do can be understood in terms of the jiggling and wiggling of atoms. And so we just take the experimental structures, wiggle them around, and hopefully we're going to learn something from that. Okay, so what are we going to do? If we want to get these things to wiggle around, we need to try to follow physics. And so there are two things that are important to keep in mind here. One is that molecular biology and chemistry is too complicated. We know a lot, like you've seen in previous talks today, about things like quantum mechanics that are really accurate for getting chemistry. We can't afford that on biology. And so the truth is too complicated. We make a lot of approximations here. But the other quote that kind of balances that is that all models are wrong. Our models are definitely wrong, but hopefully they're useful. And so I think that that's what we're trying to get out, is a model that has weaknesses. We need to know what they are, but it's still useful. And so this is sort of a quick primer on how we build our models. We've got a protein here, and we're going to calculate forces on all the atoms. So to get the forces, we need an energy function, and then we just take the kinds of things that you might learn in a freshman chemistry class and train them. So we start out with masses for our atoms, but the atoms are connected to each other. We have chemical bonds. And so we're going to need to put in some terms that link these atoms together and make sure that they don't just float off in space. So we've got bond stretching, we've got angle bending terms because there's local geometries. They take up space. So we've got van der Waals. There's things like this lysine that's negatively charged that interacts with water. We need to have water there. We've got to have electrostatics. And then there's some things that are purely quantum, like double bonds and so on, and we have to put in some terms for those. So we put all this together, we end up with a function that looks something like this. Most of the people in the field use similar functions, and it just describes all of those energies in a way that we can calculate the forces and integrate equations of motion to figure out how these things might move in time. One of the things I'm not going to talk about today is that a big part of my lab works on training these functions. We use quantum mechanics for all of our training. Some people use statistical functions. There's a lot of nice work now, people using machine learning to get functions. We've trained on quantum mechanics because my belief is that when you use statistical functions or machine learning, things like that, they tend to have trouble getting far away from the training data. And for things like dynamics and molecules, we don't have a lot of experimental data on dynamics yet. So we use quantum mechanics because it's more likely to be accurate far away from our training data. So this is an example of one kind of training that we would do where we've got a two-dimensional scan that we're doing on this little amino acid, and it's something where we can afford to do the quantum mechanics. And so we might do thousands of optimizations, get a multidimensional energy surface, and then in this case, we looked at our existing function and said, well, the minima are in the right places, but when we distort the structure like you would in dynamics or when you have molecules coming together, we don't reproduce these higher energy states from the quantum mechanics. And so a few years ago, we went through and retrained all of our amino acid side chains to get a better agreement with experiment. And we do that for all different parts of our model, constantly trying to update and improve it. And these are becoming very widely used. These are a few models from my lab. And just recently, we passed 1,000 citations in a year just to a single paper on these. And so doing this kind of modeling for biology is starting to be a very important part of understanding how biomolecules work. So the force fields go into simulation software. My group is one of the lead developers for the AMBER program. It's one of the most widely used simulation programs. This adds in all of the other things that you would need, not just integrating equations of motion, but thermostats and barostats and pH and many other kinds of methods, over 1 million lines of code in this software. We run AMBER on GPUs. This is some examples of timings. These are on a very small virus, but it's about the size of the simulations I'm going to show you today, 1 million atoms. And when we run them on CPUs, they're very small. These are nanoseconds per day in simulation. And in order to reach timescales and things that are relevant, we need GPUs where we can get up to 30 or 40 nanoseconds a day. Even that's really not quite fast enough, but it's starting to be able to reach things that are of interest. And so all of AMBER runs these calculations on the GPUs. Okay, so we have to spend a lot of computer power on this. You might ask, do you learn anything useful? There's a lot of approximations, and so we do lots and lots of validation. This is one example from our group a few years ago with our protein force field and water model, where we did protein folding simulations on proteins of different sizes and different types of structure and showed that we could pretty accurately reproduce those structures. So we do a reasonable job on structure. The other thing we want to do is motion. We want to make sure our dynamics are right. And this is an example comparing in blue and red simulation data to experiment in black. And what this is across the sequence of this small protein lysozyme, the experiment will tell us which parts are moving and which parts aren't. Things that are low here on this graph are flexible, things that are high are rigid, and we do a pretty good job matching the NMR data on that. The NMR can't tell us what the motion is, and so once we've compared to the experiment, we can say, well, now we can actually give you a model for what's going on in that experiment. And then the last thing we want is energy. Energies really are important for things like equilibrium. So here's an example where we have two proteins that are coming together in binding, maybe like ACE2 in the spike, and they form a complex. And there's a free energy change that tells you how strong that interaction is. And working with Dan Rowley's group at Stony Brook, they measured the experimental impact of a bunch of amino acid mutations. And we did the calculation of those same mutations. And you can see from this x equals y diagonal line that we do a pretty good job of reproducing those binding affinity changes. So despite all the approximations, we think we're doing a pretty reasonable job. I'm not going to get into these, but for those of you that know different simulation methods, this is just a list I made of some of the things that went in to the calculations that we're doing. We do a lot of atomistic MD and some coarse grain multi-scale models, and then advanced sampling techniques and so on. Okay, so back to our spike now. We want to simulate this, and so we need to start out from our experimental structures. And so this is an example of one from cryo-EM experiments. They give us a pretty good view of what the spike looks like, and these experiments have really come a long way in the past 10 years to where they're taking over a lot of our experimental structure determination. But they tend to have things that are missing, especially things that are flexible, and the flexible bits tend to be on the surface. So the first thing we have to do is build in all of the flexible loops that they couldn't see because they were too fuzzy, and that's everything that's in purple here. Now on top of that are all of those glycans, which are long complex sugar chains. They couldn't see those in the experiments either, and so those are the blue pieces. So even though the experiment saw the core, you can see now from the point of view of another molecule interacting with this, there was a lot that was missing. And this is one of the reasons why we don't really know how these work. And so that's what we're going to use in our simulation model. I'm showing this all as blobs, but we do build these all in full atomic detail and have full water. This is an example of the system in the simulations I'm going to show you today, where we've got our protein, we've got those sugars that are shown here in green. We include water and salt and everything we can to try to get this as close as possible to the actual biologically relevant conditions. This droplet ends up being about 22 nanometers. It sounds very small, but that's the size of a transistor on a CPU from maybe 10 years ago or something like that. So experimentalists are quite used to working with things on the nanoscale. And we're just simulating the same thing. And now that we have GPUs, we can finally start to work on the same systems that are being done in the experiment. Okay, so we do our simulation. I'm not showing all of the water and all the other details on this, and we find that not much really happens. These simulations, we can afford to do hundreds of nanoseconds of this, and we find that it just doesn't change much. So I'm going to map this onto some collective variables. We've got 3 million degrees of freedom in this system, and sort of order to understand it, we've got to project it onto some reduced space, some latent space. And so here is a structure with this blue binding domain closed and the red one is open. And we want to know, how does it open and close? So I'm going to take two variables. One is going to be the angle that makes with regard to this central axis here, and the other one is the rotation. We notice that this domain rotates when it's open. So if I project my simulations onto those two collective variables, my opening angle and my dihedral, the ones that are closed, they fluctuate a little bit, but they stay closed. This X is the experimental values. And then here we've got one with one open, like in this red state, and when it's open, it's quite flexible. It's looking around for ACE2, but the closed ones stay closed. We never get transitions, even though these are a month of simulation. We don't get transitions back and forth. So we can't learn how it actually works. What are the important parts? Are there intermediates? Things like that. And so we want to learn how it changes. So we can think about this as being on a map. We've got two locations on the map. We've got, for example, here on this one, we start out at the Red Lake Trail, and we want to get back to our Aspen Glade campground, and we want to know how to get there. We're very used to these days being able to just open your phone and map things out on Google, and it will not only tell you where you are from a GPS, but it also maps the pathway for you and tells you what an ideal path is. And so here, if we tried to just do a direct calculation, like interpolating between our experimental structures, we would end up going straight over this mountain, and that would not be a very good pathway. And so this dotted line is what our pathway mapping algorithm has told us to do, and that's following a low energy path, and that's the way we should actually go. And so we do the same thing on the computers. We use on this what's called a nudged elastic band. There are lots of these string methods, and in short, what you do is you start out where you've got two states, your initial state and your final state, and every one of these circles is going to correspond to a full simulation. Now, we map these out, we connect these with springs, and so we're doing lots of simulations. Every one of them is going to be attached by a spring that keeps them from moving too close to each other, and so they have to communicate periodically during the simulation. Then we do some decomposition on the forces. I won't get into too many of the details, but we remove our normal force field forces from in between the springs so that they can climb over the barriers. And then we run the whole thing through an optimization protocol and get out what our final pathway is. So that's what we're going to do here on this spike. We're going to map out that process of opening and closing so we not only know what our initial and final structures are, we can see where the energy basins are, have some idea how much energy it takes, and look at intermediate structures. One of the things that we noticed was that there was a pocket here in a conserved region that we didn't see in the experiments. That region was too fuzzy. And so now I'm going to zoom in past all of these surface glycans and get into the core of this where an antibody would not be able to reach and take a look and see what's going on here. We have a pocket, and now looking at some of the chemical detail, we can see that there's space here. Now we're going to close that spike back down again, and you can see not only this little tunnel, but this whole pocket itself disappears. And so that is only there in the open spike, and it's not there in the closed spike. Okay, so the next thing we want to do is we want our map to have topographic information. Like that one I showed you before, we need elevations. And so what we're going to do is project that pathway that I calculated onto a grid. And then instead of trying to run very long simulations, we run lots and lots of short simulations and just tell them, well, all you have to do is explore locally. Every one of these colors is an independent simulation. Run a few hundred of those, and then combine all of that data together, and we get out a free energy landscape. If you're not used to looking at these landscapes, this one's color-coded by free energy. Low free energy is good, and so blue here is where it really wants to spend its time. That's the global minimum. And then up here at this state where we see it experimentally being open, that's also a minimum, but it's a local minimum, and there's some barriers in between. So once we have these energy landscapes, we can start to ask questions about sensitivity of the spike to different kinds of changes. One of those, here's our wild type, is that we noticed that there is an alanine in this pocket. Alanine is a very small amino acid, and it's in this pocket, and that pocket disappears in the closed state, and there's no space for anything larger. And so we thought, well, if we make that bigger, if this pocket is real, it shouldn't be able to close anymore. And so we did. We substituted with a valine, which has got 2 extra carbon atoms. It's very small, recalculated all those free energies, and you can see now the favored conformation of the spike is the open form. It doesn't close anymore, and that's because we created a steric clash in the closed form. And so that showed us that this pocket actually did control this exposure of a receptor binding domain, even though it was very far away. It's what we call an allosteric pocket. And then interestingly, we found out that this A522V actually is seen in patients in the clinic. And it, as we would have predicted, it increases antibody neutralization. It's easier for the antibodies to get to this because it doesn't close as much anymore. And so that was consistent with our prediction. Why the virus would want to have something that's easier to neutralize, we don't really know yet. My hypothesis is that there were other changes in the spike that made opening difficult, and those are the changes that it wanted to make, and now it had to compensate. And so it did this mutation to compensate to help it open it back up again. One of the problems with studying these in experiments is that when we make changes that make the spike more flexible, it tends to be very hard to purify. They just kind of turn into gunk and stick together, and so we can't really study this very well experimentally. And so what we think a better approach would be to use this pocket and develop small molecules that we could make the spike, purify it, and then add those molecules in to shift around the spike and say, well, what does that have? What impact does that have on the opening and closing? And so working with Rob Rizzo in applied math that many of you probably know, he's the lead developer on a program called DOC for virtual screening. We gave him this pocket and he screened libraries of compounds and found ones that make good contacts and look like they fit into that pocket very well. And then we're able to take those and do simulations. On the left, we don't have the molecule bound, and the right, that molecule is in pink. And when we run our simulations, the one on the left with no molecule, it opens sometimes, but it definitely closes and prefers being closed. The one on the right has trouble closing because that small molecule has gotten in there and now it's kind of wedged into the hinge. And so the free energy calculations show the same thing. Here's the one I showed you before for just the spike by itself. And now we haven't made any mutations to it, but we've added that small molecule and it had the effect of shifting this so that it's very difficult to close. And so we're working with experimentalists to screen some of these molecules, thinking that they will be useful for being able to probe exactly what's going on with this spike opening. Okay, now we've started to move more into trying to really understand the trigger. We think we know how it opens and closes and why it does that, and what spots might be changing it. But what we'd really like to know is what sets off this triggering mechanism. We know here on the left, this S1 subunit, the mushroom cap, that's the part that recognizes the receptor. And so it's going to bind to ACE2 here. But this part in the bottom, this is the harpoon that's going to pull these two cells together, or the virus and the host cell. How are they connected to each other? We don't really understand this link. Why is it that binding to ACE2 leads to all these conformational changes? Even though many, many people have studied this and know that this happens, we don't know why. And that could be very important to trying to block this process. Okay, well, the most straightforward explanation, like we've drawn out here in our models, is that once you bind to ACE2, it just kind of pulls on that cap and pulls it off. They're not connected anymore. That's kind of part of a longer story, but these are separate proteins. So you could, in principle, pull this top off, leave this behind, and then it'll rearrange. So once that S2 is by itself, people think, okay, it's unstable and it's going to start to go through some changes. But again, those are models. We have no experimental structure information, and we don't know if that happens. So there are two different experiments that have been reproduced many times now that conflict with each other. And this really highlights how we don't know what's going on with this virus. The first one is that it turns out we don't have to bind to ACE2 in order to get this process to happen. And so this is a paper last year from Science, and they showed that those S1 subunits, that mushroom cap, that comes off on its own sometimes in the experiments, even when there's no human cell around and no ACE2 around. It just happens on its own. This one on the right, same thing. These are pictures of viruses that have never been exposed to ACE2 or any other cells. And most of the spikes on the surface are in this sort of needle-like post-fusion thing where they think that they've already attached themselves. Once they're in this post-fusion state, they're inactivated and it's irreversible. We can't go back. One of the things we'd like to do is figure out, well, if this is happening on its own, is there a way that we could get it to happen? Because if we understood it, maybe we could poke it in the right way and all of those subunits would come off and then the virus wouldn't be able to get into the cells anymore. We have no idea how that happens. We just know that it does. So we don't need ACE2 around. And then the other thing that's odd is that even when ACE2 is around, we can bind ACE2. These are experimental structures. We can bind an ACE2 to every one of those receptor binding domains, and it apparently has no effect. And so even though we say, oh, well, it binds to the host cell and then the cap comes off, that doesn't seem to be how it works, not from these experiments. So what is going on here? Well, we think what's happening is that these experiments where we see all of these things bound to them, they're using a variant of the spike that's called the 2p spike. Now the 2p spike, it's got a substitution in it and it ends up being very important for all of you personally. This is our pre-fusion structure here where it's got that cap on it. That's the part that's going to bind to ACE2. And here's our post-fusion experimental structure. Let's get rid of the cap and say, one of the big differences here is that these helices in the middle are much, much longer. Parts of this that are folded down rotate upward and they make these long central helices. Those are like the harpoons that stick out and try to attach themselves to the surface. So some extremely clever people figured out that, and if you know things about proteins, you know that if you put a proline amino acid in, it's a very unusual amino acid with a ring on the backbone. Those do not like being in helices. And so they put two prolines in a row. P stands for proline, and so this is the 2P. Two prolines in a row, and all three of these, and that doesn't happen anymore. And that makes it stable in the pre-fusion structure, and that is in every one of the vaccines. And this is huge because otherwise, like from those experiments I showed you, it would end up falling apart and being the post-fusion state and your antibodies would recognize the wrong thing. So every one of the vaccines, at least that we have in this country, have this. And I really love this quote from this paper from, well, this is a recent news article, but the paper that described this was back in 2017. And they said, this should be advantageous for the development of vaccine candidates against emerging coronaviruses. They were completely right, and it is in every one of the vaccines, and figuring this out made a huge difference in the world. This is impact on science, and the person who did this, Jason McClellan, is going to be here in a couple of weeks, and I really encourage you to go and see him and see this talk because he is the reason that you have a vaccine right now. Okay, but the 2P has another problem, and that's because it's so easy to work with, all of the experimentalists have started using it. Really nice, easy to purify, easy to work with. And these experiments show that when you open the spike, so closed here on the left, open on the right, this is a 2P, like all of these experiments with multiple ones open. The core part, that part that's supposed to release those torpedoes, or the harpoons, nothing has happened. There's no change. And so this is why people have hypothesized, oh, this whole piece needs to come off. And we just don't know yet how that happens. And we don't think that's right. We think, from studying the structure, that there's a little bit of a loop region in here. This is very odd when you think about protein structure. This loop interrupts this helix that's coming up. It loops around, and then it comes up again. It's in every one of the coronaviruses, and we don't know what it's there for. So based on our simulations, we think that this is the trigger and that people have missed it because they're using variants right here that stop this trigger from happening. And so I'll show you how we think this actually works, even though, again, it's a hypothesis. We don't have any experimental confirmation yet. Here's our spike. The S1 subunits are going to open up as if they would bind to an ACE2. And we're going to zoom in and do our simulations. And what we think is happening, now I'm going to get rid of that one so you can see inside, is that that loop allows a conformational change in the core. And it's not seen in any one of the experiments, but it makes a lot of sense once we start to build the models on the computer. We form lots of interactions here that are extremely stable and give off a lot of energy. And we think that this is the energy source for forming these harpoons. Okay, so on the left here is our pre-fusion structure from experiment. What we think is happening is that this little bit of helix and this loop, the loop is there because we're going to rotate this up and that loop will keep that part connected. And so it's just a little bit like a release of tension. We still stay tethered even though we can make this. And when we look at our simulation model and the final post-fusion structure, when we've lost S1 and all these other changes have happened, all of these interactions up at the top that give us so much energy in our simulations, those are also present in the post-fusion structure. We just think they happen earlier. We think they're the driving force to this change. We don't think that they're the result of the change. So why would that matter if we open up this little bit of helix? It seems like a small change. So we flip this up. We've got a loop that unfolds. And looking down now on these helices, this is what we think the change is. This out here on the outside in purple, that's our S1 part that needs to actually come off. And in the simulations, once we create this intermediate, you can see this interaction here between opposite charges of amino acids that just spontaneously breaks off. And there's a lot of details that we have that won't get into that explain why this part rotates and twists itself off of the mushroom cap. Once that happens, the S1 can come off, and now we've just got that S2 behind, and it's going to release those long chains that are going to insert themselves into the host cell. And at that point, it's attached, and there's very little that you can do. Okay, so just to summarize here, the experiments have been wonderful. We get a view into this virus over the past year that we've never had on coronaviruses before. And they've given us a lot of information about the spike, but they don't really tell us how it moves and a lot of the details that we need to know. The simulations are able to take that and add into it. I didn't mention, but we've learned a lot about why those glycans are there. They're very important. It helps the virus to hide, but they also have a structural role. We can explain some of these variants, even ones that are far away from antibodies bind, and it would be very difficult to understand why they would change the behavior. But we understand that now through the energy surfaces. We're getting some ideas how we can control the opening. And then I think most important is that if we figure out how this machine actually works, that may give us a way that we can go in and try to trigger it on our own, the way that we see it happen spontaneously. And that's a conserved mechanism against all coronaviruses. And then most important, this is just me on my little soapbox, is that we need to shift now from worrying about the pandemic and things like that to really focusing on getting people vaccinated, because we are still letting this virus evolve. So far, it's been evolving without having evolutionary pressure from the vaccines. As more and more people are vaccinated, then if we let it keep evolving, it is more quickly going to overcome that immunity. And we really need to get people to understand that the vaccines are safe, that they're effective. There may be very small risk of side effects, but that this is something we need to do, not just to protect ourselves, but to keep it from evolving and changing. I want to thank all the people in my group, especially Lucy, Kellen, and Yuzhang, who were the lead grad students on this project. We have lots of undergraduates that help out with all of our data analysis, and experimental and computational collaborators, Remy Amaro's group and Arvind Ramanathan. They were on the team that we got the Gordon Bell Prize with, and the COVID-19 HPC consortium for all of the GPUs that we use. And I'd be happy to answer any questions.
Carlos, thank you very much indeed. Yeah. Some questions from the audience?